so I'm going to get started. Welcome, everybody. We have people from 5 continents who have signed up for this webinar. We're really excited. So greetings to all of our participants from all over the world. I'm Jean Greenberg, President of the International Society for Molecular Plant Microbe Interactions. Today marks the exciting first installment of our Road to Jeju Rising Stars in IS-MPMI webinar series. Just a little bit of news. Our new website has officially launched as of July 1. So you can customize your membership information, and we're going to stand up the job board, and community bulletin events have already started to come in. If you want to add something, please go ahead and do that. Many of the 2025 Congress presentations are there that you can watch freely on our website. And this is your hub for all things IS-MPMI. So you can visit us at the website listed there. Let's see. I'm going to introduce Maurizio. He is the main organizer, together with his committee, of this series.
Thanks for the introduction, Jean. And yeah, I'd also like to welcome everyone. It's wonderful to see so many people actually online here. So for people who attended the previous webinar, you might already know this is part of our Road to Jeju webinar series. And the Rising Stars of IS-MPMI is part of this Road to Jeju journey. So as you may or may not know, in July of next year, the next IS-MPMI Congress will take place in Jeju Island, South Korea, from the 18th to the 22nd of July. So that's kind of our main flagship meeting of the society, right? And that usually tends to bring together more than 1,200 researchers from around the world to share the latest in MPMI. So actually, if you haven't already, mark your calendars for those dates. But to build momentum on the way to the Congress and to keep our community engaged globally, we're running these webinar series every 6 to 8 weeks from now through to May 2027. And it alternates between 2 formats. So the first format is Legends of IS-MPMI, and that features in-depth talks from leaders who've shaped our field. And the second format, which is taking place today, is Rising Stars of IS-MPMI, which is meant to be a showcase for outstanding early career researchers who represent the future of our society and of our community. So our first Legends webinar happened a few weeks ago, actually, with a great talk from Roger Innes. And actually, in case you missed it, you can check it out on our YouTube channel or on the IS-MPMI website. You can use the QR code on the screen right now. And as you already know, every session is free, open to the public, and held on Zoom. If you or anybody that you know misses this, you can always find the uploads on YouTube and, as I said, on our website. So please help us spread the word on this webinar series. So for the Legends of IS-MPMI, here's the lineup, and there's actually an exciting piece of news to share. So as I said, we opened the webinar series with Roger Innes, who gave a fantastic talk, and the talk and extended Q&A actually. So we did both the talk and an extended Q&A session to address the questions that could not be addressed during the talk. These are both up on our YouTube channel and website. And looking ahead, we have a very exciting talk coming up from Jonathan Jones from the Sainsbury Lab in Norwich, UK. And we have a new speaker that has been added to the lineup. So this is Eva Kondorosi from the Biological Research Center in Szeged, Hungary. So Eva, for those of you who may not know her, is a pioneer of nitrogen-fixing symbiosis, and she will be giving a talk in October. Then in January, we have a talk from Jane Parker from the Max Planck Institute for Plant Breeding Research in Cologne, Germany. And then we'll close out the series in May with Xinyan Dong from Duke University. So some of the exact dates are still being confirmed, so we will announce those soon. So these are 5 genuine leaders of our field throughout the next year, and we hope that you will join us for all of them. So for the Rising Stars of IS-MPMI, so this is actually today's the very first issue of this webinar format. So this, as I said, this is a session dedicated to highlighting the next generation of our field. It's a showcase for outstanding early career researchers. So this means PhD students and postdocs who are IS-MPMI members. So anybody who fits that description is invited to self-nominate, and our board selects the speakers based on scientific merit and based on contributions to the MPMI community. So there will be 3 of these Rising Star sessions across the series. The first one is today. The next one will be in November 18th of this year, and there will be a third one in March 2027. So actually, nominations for the second session will open in September 1st and close on October 14th. So if you or a student or postdoc you know would like to present, please keep an eye out for our announcement of nominations or self-nominations opening. And actually, there's real recognition attached beyond being able to share your research with the global IS-MPMI community. From all the Rising Star speakers, the board will select 2 awardees. So the first one will be the Get Genome Rising Star awardee. So this awardee will give a named plenary talk at the Jeju Congress next year with free registration and travel support courtesy of Get Genome. And we also have the Tanon Excellence awardee who will receive free registration and travel support to attend the conference in Jeju courtesy of Shanghai Tanon Life Sciences. So one important point that I do want to raise is that I did say that self-nomination is open to student and postdoc IS-MPMI members, but if you are an early career researcher based in the Global South, you can apply to self-nominate regardless of membership status. And actually, if you are selected, you will receive a free 3-year IS-MPMI membership. So we particularly encourage participation from Global South scientists for this webinar series. So please nominate yourselves for a second round and encourage your students and postdocs to do the same. So when the call opens, you'll be able to sign up through a form that we'll share. And if you can't access the form for whatever reason, you can always just email contact@ISMPMI, the email address on the screen here, and we'll send you a form for you to nominate yourself. So over to you, Jean.
Thank you. I want to thank our sponsors. This series is made possible by 3 major sponsors shown here. Use the QR codes or the website information if you want to learn more about them. So we are very happy today that James Canham, who is part of Get Genome, is here with us, and I think he's going to. Do you have slides, James?
No, I don't have any slides, but I can say a few words.
Okay. Go ahead.
Thank you. Yeah, thanks, Jean. Yeah, so at Get Genome, our mission is to empower researchers by making genomics more accessible. And so we're especially proud to support early career researchers as they shape the future of the field. So it's a really great pleasure for us to be supporting the rising stars of IS-MPMI. This initiative is really wonderful, and it's so fantastic to hear so much support given to Global South researchers as well. Thank you.
Okay, we'll hear back from Maurizio now.
Yeah, so talking about Global South scientists, so I want to take a moment to share something that the board is really excited to share with you guys. I mean, we've been kind of advertising this on our social media, but you may not be aware of this. So we realized as a society that our colleagues from Africa, Latin America, and the Middle East, and lower and middle income countries across Asia are significantly underrepresented in IS-MPMI. So we want to change that, and we are putting concrete steps behind this. So what we're offering is, first, if your primary affiliation is in a low-income country, IS-MPMI membership is now completely free. And secondly, if you're based in the broader Global South, you can join at 50% of the standard rate. And the third thing that I'm especially excited to mention is that our president, Jean Greenberg, is personally covering membership for the first 25 PhD students and postdocs from the Global South who request a waiver. So this is the president/membership initiative. So it's a free 2-year membership, and this is on a first come, first served basis. Just email contact@ismpmi.org. The email address that I mentioned earlier. You can find it also you can see it on the screen here, actually. You can find it on our website. So we've given away quite a few of these President's Flash Membership Initiative waivers, but there's still quite a few left. So if you're interested, do reach out ASAP. So the bottom line is that wherever you are in the world, there's now a place for you in this society. And actually, please do help us get the word out of this initiative so we can get more people involved in IS-MPMI. So
Thank
back to
you.
you, Jean.
And I'm excited to sponsor the students and postdocs. So please apply if you meet the criteria. I also want to mention that we are still raising money for our travel fund, and we have raised we have a goal of $40,000. We've raised almost 26,000 so far, and no amount is too small. So even if you have an extra $5 or $10 for the students on here, please consider contributing so that we can send as many people as possible to the meeting, or at least partially defray the cost. And also, if you're on this webinar and you're not a member of ISMPMI, please consider joining. Your membership ensures that we can cover our basic operating expenses. Today, I'm excited to share with you that previous speaker in our early showcase webinar series from 2025, Abby Rogers, is here, and she is going to moderate the session. She's just gotten her PhD, freshly minted from Purdue University, and has worked on effectors of Ralstonia, and I'm sure I'm going to mess this up, Filicorda matus, and she's about to go to Michigan State to start her postdoc, and she's been a great member of our society. So if you want to learn more about her, there's her LinkedIn page, and I'm going to hand this over to Abby.
All right. Hi, everybody, and welcome again to the first session of the Rising Stars webinar series. I'm very pleased to be helping out and moderating this series. I really love MPMI events, so very excited about this one. So for our first speaker today, I'd like you to all join me in welcoming Dr. Ni Nguyen, who recently began her postdoctoral work in Dr. Kei Haruma's lab at the University of Tokyo. And this is actually the same lab where she also recently completed her graduate studies. So today she's going to be sharing some of her work related to microbe-mediated plant nitrogen acquisition, some of which is available in her 2025 preprint and some of which will be new to the public today. So, okay, Ni, whenever you're ready, you can go ahead and take it from here.
Sorry. Thank you, Abby, for the great introduction. And my name is Ni, and today I would like to talk about plant growth promotion by endophytic fungi, where hyphae act as conduits for nitrogen and beneficial bacteria. So, as you all may know, nitrogen is a crucial macronutrient for plant growth, and its deficiency cause significant growth inhibition and yield loss. However, nitrogen deficiency is very common in nature. So to cope with this, plants form symbiotic interaction with beneficial fungi, whose hyphae network grow in soil as root extension and support plant nitrogen acquisition. So therefore, beneficial fungi have great potential for both basic research and agricultural application. However, several challenges remain. First, although fungal symbionts are well known for support plant nutrient uptake, the underlying mechanisms are still unclear, particularly the fungal genes involved. This is mainly due to the technical difficulties of genetic modification of symbiotic fungi that we have been studied so far. Secondly, the effectiveness of beneficial fungi in the field may also be influenced by the soil bacterial community. However, how these fungi interact with bacterial communities and whether this interaction also benefits the plant remains unclear. So in my talk today, I would like to provide some insights into this question using the endophytic fungus called Colletotrichum tophiidae, or from now I will call CT. This fungus is known to colonize the root of the model plant Arabidopsis thaliana and promotes plant growth under low phosphorus condition by transferring phosphorus to the plant. More importantly, CD can be genetically modified. So CD can be a potential role model for mechanistic investigation of plant and fungal symbiosis. We also found that the symbiosis between Arabidopsis thaliana and CD also extends to low nitrogen condition, where CD promotes plant growth significantly and specifically under low nitrogen condition. But not only boosting the shoot growth, when we look at the plant transcriptomic response, we also found that genes that were transcriptomically increased by the nitrogen starvation were reduced in CT colonized plant. These genes are involved in response to stimuli or stress, starvation, or senescence. So this suggests that CT-treated plants exhibited reduced stress response under nitrogen limitation. Considering that CT can transfer nutrients to the host as in our previous study, and that CT can alleviate plant nitrogen stress responses, we hypothesize that CT can transfer nitrogen to the plant. To test this hypothesis, we conduct this 15N transfer assay using 15N labeled nitrate and supply exclusively to the fungal compartment. CT fungus will be grown in this small compartment until they reach to the large compartment in here. And then the small compartment that contains 15N will be removed, and the plant will be transferred to this large compartment and grow for 20 days before harvesting the shoot and measure the 15N atomic percentage in the shoot. So as you can see with this setting, where plants were not co-incubated with the 15N sources, we ensured that the increased 15N detected in the plants was likely transferred by the fungal hyphae. And indeed, we detected a clear increase in 15N atomic percentage in CT4 treated plants. And more importantly, this increase was more pronounced under nitrogen deficiency. So yes, CT indeed transfer nitrogen to the plant, but what mechanisms underlie this transfer? To understand this, we need first to understand how fungus acquired and utilize nitrogen. So fungus, like CT, can utilize nitrate in the environment by this pathway, which was regulated by two main transcription factors called NIT4 and RAA. This pathway, however, in the presence of the preferred nitrogen source for the fungus like ammonium or glutamine, was mostly deactivated. We found that under our low nitrogen condition, the transcription factor NIT4 and the nitrate reductase was induced, suggesting that this pathway in CT was induced and likely activated. So, does this pathway contribute to CT-mediated nitrogen transfer and growth promotion? To investigate this, we use the two mutants that are NIT4 and RAA mutants, which are impaired in nitrate utilization as their growth in the media containing nitrate as the main nitrogen sources drop significantly compared to the wild-type CT4, especially the RAA mutant, which show severe impairments. When investigate the nitrogen transfer, we observe a significant drop in RAA mutant, which can later be rescued by the complementation of fungal CT-RAA. Meanwhile, the NIT4 mutant retained a similar nitrogen transfer to the wild-type CT4. This result suggests that the fungal transcription factor RAA is crucial for CT-mediated nitrogen transfer. And also consistent with the previous nitrogen transfer, the plant growth promotion in this RAA mutant also drops significantly while retaining in the NIC4 mutant. This further confirms the important role of this transcription factor RAA in nitrogen transfer and growth promotion. So in this section, I presented our finding on this fungal transcription factor RAA. And to my knowledge, RAA is the first fungal gene identified as being involved in nitrogen transfer during plant fungus symbiosis. So as I mentioned from the beginning, another challenge that we have when studying plant fungus symbiosis is the soil bacterial community. So our next question is to ask how does CT interact and modulate the rhizospheric bacterial community. This question was partly raised by our observation in our soil pot assay, where CT significantly enhanced plant growth and fitness under this condition. And on those not presented today, we also conducted field trials throughout different seasons and confirmed that CT consistently enhanced plant growth under non-sterilized low nitrogen soil. And in some cases, this plant growth promotion may even exceed what we observe in sterilized agar conditions, suggesting that CT may also cooperate with the root bacterial community to further enhance plant growth. Indeed, we observe a significant shift in the bacterial community in roots of CT colonized plant under non-fertilized low nitrogen field, and found that specific bacterial taxa were enriched in CT colonized roots. So the next step to understand how the bacteria that co-colonizing the roots with CT may influence this symbiosis, we isolated the bacteria from CT colonized root and successfully recovered this bacterium, these isolates that belongs to the Parabacoderia genus that are known that have many species known for plant growth promotion effects through nitrogen fixation, oxime production, phosphate solubilization, or plant protection, etc. And these isolates can in fact promote plant growth under low nitrogen agar that mimic the soil environment by amino acid supplementation. We named this isolate as Pantan, and it belongs to the Parabacoderia monticola species. Also, as you can see from this graph, our isolation also resulted in other bacterial taxa that were not enriched by CT. Include this isolate, which we call as ACA, belong to Acinetobacter gluei. And in contrast with Pantan, this isolate inhibits plant growth. And interestingly, we observe that while Pantan can grow along the fungal hyphae, ACA did not show such growth, suggesting that CT hyphae serves as conduits for the beneficial bacteria Pantan, but not for ACA. This also raised a question whether Pantan can move along the fungal hyphae, as some bacteria are known to ride on fungal hyphae. So to test this, we generated Pantan GFP lines and confirmed that Pantan moved along the fungal hyphae, as you can see from this video footage. So Pantan approached the roots through CT hyphae. Then, how can Pantan promote plant growth under low nitrogen condition? We analyzed the genome of the bacteria that we isolated and found that Pantan genome is enriched in amino acid metabolism genes more than other isolates that we have, and consistent with its ability. This also consistent with its ability to boost plant growth on its own or together with CT, specifically in the presence of amino acids, while such plant growth promotion was not observed when no amino acid was added, suggesting that amino acid metabolisms may be crucial for PAM10-mediated plant growth promotion. And we confirmed this by knocking out a single copy gene in PAM10 that is responsible for valine and leucine metabolism, called ILVE. When grown on media which contain only valine and leucine as the amino acid sources, the disruption of ILVE not only impair PAM10 growth, as you can see from this photo, but also its plant growth promotion effects, which can be complemented by the addition of another amino acids. So this result confirming that LIVE is essential for PAM10 valine and leucine metabolism. And this metabolism plays crucial roles in PAM10-mediated growth promotion. So to summarize my talk today, I introduced Colletotrichum tophiidae fungus as a fungal symbiont that promotes plant growth under low nitrogen conditions. I also presented the underlying mechanism, including nitrogen transfer to the host plant, which is regulated by the fungal transcription factor RAA. In addition to that, CT also preached the beneficial bacteria such as Parabaccorderia monticola PAM10 to the roots. And this beneficial bacteria can further promote plant growth by metabolizing amino acids in the environment and diversifying the range of nitrogen sources that are available to the plants. So throughout this study, we established Arabidopsis, CT, and PAM10 system as a model where each of the partner can be genetically modified. And this may pave our way for investigating the mechanism underlying the symbiosis, particularly interkingdom symbiosis. Also, our finding, which also includes field trial, highlights the potential of CT to enhance plant growth in field conditions while reducing fertilizer inputs, which is becoming increasingly important for sustainable agriculture. So with this, I would like to conclude my talk and thank everyone who made this research possible. And I'm happy to take any question. Thank you.
All right, thank you very much, Ni. So it looks like we do have a question. So Pawan Kumar has asked, how are the PAM bacteria and the CT fungi interacting in the absence of host plants?
Thank you for your question. This is a great question. So we also try to co-inoculate CT together with this bacteria without the host plant. And actually, the video footage that I showed here is without the host plant. So you can see that even without the host, PAM10 can ride on the hyphae and grow along the fungal hyphae.
Okay, excellent. And then we also have another question from Ved Prakash, who has asked, what could be the possible reason that CT promotes PAM10 colonization, but not colonization of the pathogenic bacteria that you mentioned?
Thank you. This is a great question. So we are now still trying to understand the mechanism of this, but it is also known that not all bacteria can ride on the fungal hyphae. So the fungal hyphae itself may select which bacteria can ride or cannot ride on its hyphae. We don't know the mechanism on our CT so far, but on other fungus, this mechanism may include that the bacteria provides some cues to the fungal hyphae. For example, in that case, bacteria can provide thiamine to the fungal hyphae. But in CT, we're still trying to figure out what is the mechanism for this. Thank you.
Okay, and then I actually have one question, and then I think if we don't have any other questions from the audience, I think that's it. So in the presence of the host plant, do you see PAM preferentially colonizing the hyphae, or can it colonize the host's root tissue without the presence of the hyphae as well?
Sorry, may you repeat the question? Sorry.
Sure. In the presence of a host plant, do you see this PAM-10 colonizing the root tissue itself, or does it preferentially colonize the fungal hyphae?
Well, that is very great question. Currently, our experimental setting does not allow us to quantify and separate. And so in order to know which one PAM10 actually prefer, we need to have a way to quantify PAM10 colonization on roots. That should be easy, but on hyphae. So currently our setting does not allow us to quantify this preference, but that is a very interesting question. Thank you.
Okay, Ni, and then we have one last question from the audience. So Atira Mohandas has asked, does CT induce a cytosolic calcium elevation in the plant upon perception?
So specifically for that response, we did not see a very strong or clear response from our RNA-seq data. We can see in general, in order to form symbiosis with CT, plants would have certain response that related to stress or to interact with fungi in general, but we did not look into that response specifically. Thank you.
Okay, thank you very much. Okay, thank you very much for that presentation. So next, I would like to welcome Dr. Irouj Fatima, who is a postdoctoral fellow at the KAUST Institute in Saudi Arabia. Dr. Fatima completed her PhD in India at the National Institute of Plant Genome Research prior to joining Dr. Simon Kertinger's lab, where she currently works on understanding cereal genetics and genomics. And today she's going to tell us a bit about the binding of a lectin receptor kinase, which leads to a non-host resistance response. So go ahead whenever you're ready.
Hello, everyone. Thank you so much, Abby, for such a nice introduction. So today I'm going to talk about conserved rust fungal ligands that trigger non-host disease resistance in cereal crops. And of course, for the detail, we have recently pre-printed this in bioRxiv, and you can get more detail on this. I would like to start my talk. So I'm gonna talk about. So basically, I will start with the finding of my study. So this study identified the first molecularly resolved ligand receptor interaction contributing to non-host resistance in cereal crops. And here we identified the orthologous glycoside hydrolase as a fungal ligand that is perceived by barley receptor kinase. And the barley host status to leaf rust is influenced by the direct recognition of fungal GH5 by a lectin receptor kinase. However, if you look at the host interactions, so to infect the barley, adapted rust species must evade this recognition. And with this clue, I would like to start with the background. Cereal rust are a problematic creature. However, if you look in the microscope, they're a beautiful creature. But the problematic side is the high level of host specificity, high adaptability, and there are a lot of efforts going on to have a disease resistance lines, but they are often overcome by the emergence of highly virulent rust strains, and that could also lead to severe epidemics. To solve this problem and to overcome the challenge, we employed the non-host resistance, that is a resistance shown by entire plant species to all isolates of pathogens. And for this, a beautiful example and excellent model is the wheat and barley. So wheat and barley, they diverged 12 million years ago. And if you look at the barley, the domesticated barley and the white barley, they diverged 8 million years ago. And if we look at the pathosystem, the Puccinia hordi is a host for the domesticated barley, but wild barley and wheat are the non-host for this pathogen. And Puccinia hordi bulbosi is the host for the wild barley, but domesticated barley and wheat are the non-host. And then the wheat leaf rust, of course, is not able to infect the domesticated or wild barley. So to sum up, I would like to say the limited host range of most of the pathogens suggests that these non-host disease resistance mechanisms are not easy to overcome, and the non-host resistance could be a path toward durable resistance. Yet it is poorly understood. If you look at the literature, 60 years ago, the research started, and till date, only 5 NHR genes have been identified compared to more than hundreds of host-resistant genes. Our group has cloned 2 genes encoding lectin receptor kinases that is involved in NHR, one from the domesticated barley, RPHQ2 locus, and another from the wild bulbous barley, RPH22. When these genes were transferred into the highly susceptible golden suspicious barley plants, then they offered the resistance. For example, in case of the domesticated barley, the RPHQ2 offered resistance to the Puccinia hordi bulbosi, wild barley leaf rust, and the wheat leaf rust, the non-adapted pathogen. And when the RPH22 was transferred, it offered resistance to the wheat leaf rust and the domesticated barley leaf rust. And this gene encode for the L-type lectin receptor kinases. And if you look at the lecarcase from domesticated and wild barley, they have 85% amino acid identity. Basically, these lecarcase act as a sentinels. Adapted rust evade the perception, while the non-adapted rust perceived by lecarcase and they trigger HR. So with this information, I have 4 important questions to answer during my research. And with this story, we were actually able to do that. So first, the resistance outcome is being governed by which domain? So is it the extracellular lectin domain or the kinase domain? Second, what is that rust ligand that is recognized by lecarcase? The third question is whether this lecarcase-mediated resistance can be functionally transferable across the species. And fourth, the most important, is the recognition of the non-adapted rust ligands by lecarcase, a conserved mechanism. So to answer these questions, we need a transient system. Of course, we cannot do everything with the barley transgenics. The implanter responses that we observe, the resistance here, for that we need a transient system. For that, we established that in our lab where we isolated the apoplastic fluids from the sospitrid infected with pathogens, and then we used it as a source of ligand, and then we expressed the lecarcase in bentamiana, and then score for the HR for the ligand, basically ligand perception, it will trigger the HR. With this transient system, we were able to recapitulate the implanter responses mediated by lecarcase. As we can see that when the non-adapted pathogens, the apoplastic fluids from the non-adapted pathogens were given as a treatment to the HP lecarcase, which is expressed in bentamiana, it triggered cell death response. And in the case of HP lecarcase, also the non-adapted pathogen perception triggered the cell death response. After this, we were interested to know which domain is actually responsible for the perception. So for this, we did the domain swapping, where the lectin domain swap of domesticated lecarcase with wild lecarcase was swapped, and it started recognizing the adapted version, that is from pH. And when the lectin domain swap of the wild lecarcase was swapped with the domesticated version, it started the another adapted version, that is a PHB. After this, we were also interested to know what is the source or what is the nature of this ligand. So we treated it with heat, lipase, and proteinase. And interestingly, we found that in case of heat and proteinase treatment, the HR was completely abolished. Now it's time to answer, it was the time to answer the second question, that is, what is this rust ligand? For this, we had a beautiful pipeline, and the search began with the same apoplastic wash fluid that we had it from the sospitrid plants, and then we did the LCMS. We didn't have information for the genome and the transcriptome for this pH and PHB isolate that we were using here in the lab. We generated that so that it can be serve as a library. Then we came up with 232 apoplastic rust fungal proteins. We ran them through AlphaFold, and based on their ranking, we came up with 40, which we tested transiently and scored for HR. And with this pipeline, we were able to identify glycoside hydrolase GH5, which is predicted to have a beta 1,6 glucosidase activity. And this we then tested with actual candidate and did all the transient assay by expressing the LEC-RK with GH5. When we expressed the pH GH5 with HB LEC-RK, we could see the cell death response, however, not in the case of HB LEC-RK. And when we expressed the PHB GH5 with HB LEC-RK, it triggered the cell death response, but not in the other case. After that, the next important thing was to test the interaction. For that, we did the co-IP, and our co-IP results clearly indicate that the PHGH5 co-precipitated more strongly with HB-LEC-RK than PHGH5. However, the HB-LEC-RK displayed a strong interaction for PHB-GH5 than PHGH5. With this, we also wanted to, since there is a difference in the interaction strength, so we wanted to validate it with another assay. We established the hybrid-based quantitative co-IP, and we were able to validate the same findings with this assay also. So with the co-IP, we were able to see the interaction. We wanted to also know whether this interaction reflects the direct binding, for which we did the binding assays using hybrid LGBIT complementation system. Basically, this is a kind of a saturation binding assay where we saturated with GH5 concentration, and this binding assay actually revealed the difference in the binding affinity, as we could see that PHGH5 bound HB-LEC-RK with higher affinity, that is kind of 50 nanomoles, compared to PHBGH5 that was something around 1.79 micromole. And conversely, we also saw that HB-LEC-RK displayed higher affinity for the PHBGH5, whereas binding with the PHGH5 was weaker. With this information, we also wanted to validate in planta. But the bottleneck is that we cannot modify Rust are recalcitrant for any modification. For this, we moved to the another pathosystem, that is rice blast pathogen, Magnaporti, and we expressed these GH5 variants in this Magnaporti. So basically, Magnaporti expressing PHGH5 triggered resistance, basically HB-LEC-RK triggered resistance towards the Magnaporti expressing PHGH5, and the Magnaporti, when the Magnaporti is expressed with PHBGH5, we could see the resistance response in the HB-LEC-RK transgenic barley lines. Now, with this, it was a time to answer the third question, whether this LEC-RK mediated resistance can functionally be transferable across the species. And to answer this question, basically, we know this Puccinia triticina is kind of a non-host. When HB and HB-LEC-RK transgenic lines were used, the Puccinia triticina cannot grow well. And we could also saw this in our transient assay, where we used the GH5 from Puccinia triticina, wheat leaf rust. It triggered HR for both barley LEC-RK. So we transferred the barley LEC-RK into the wheat, and we could see the increase in resistance against Puccinia triticina. And this we further validated in our field trials for 2 seasons, where we could actually see for domesticated and wild barley LEC-RKs, the transfer of these genes into the wheat cultivar fielder provided resistance against mix of 7 different Puccinia triticina isolates. Now, the most important question is the recognition of non-adapted Rust ligand by LEC-RK a conserved mechanism. For this part, a PhD candidate in our group, Natalia, she did a beautiful analysis where she did the phylogeny for 7 Rust species and identified that PHGH5 and PHBGH5, they are the member of the highly conserved clade. Following this information, we were interested to explore the possibility of expanded recognition. of the GH5 by LEC-RK. So we move to the another system where we tested the multiple combination of RICE, LEC-RKs, and GH5s in our transient assays. So in this case, we could see the expanded recognition spectrum where different LEC-RKs could trigger the cell death response for the barley leaf rust and also for the one of the interesting thing was it was for the stem rust also. And one of the LEC-RK that is OS6 LEC-RK, it triggered specifically for the stem rust GH5. And then we decided to test whether it could work if we transfer this LEC-RK into the wheat line, whether it could provide resistance. And actually, we can see that this is when the stem rust infection was provided into the wheat, it could not grow well. And we could see that these transgenic line provided resistance towards the stem rust genes also. So with this, the story has a two key message. First is the recognition of non-adapted rust by LEC-RKs is a conserved mechanism. Second, this conserved NHR mechanism provides a foundation for engineering the LEC-RK variants, which can have a broadened recognition of specificities across multiple crops. And now, finally, I would like to thank and acknowledge all the support provided by Simon, and also the special thank to Natalia, the PhD student, and all the members, and a special thank to the IS-MPMI board member and the selection committee for giving me this opportunity to present my work. And thank you all for listening. I'm happy to take the questions.
All right, Aruj, very good talk. So it looks like we have a couple of questions right away. So Pawan Kumar has asked, why use apoplastic fluid for identification of GH5 and other proteins? Could you tell us something about disease progression of these rust fungus in hosts and non-host plants?
Okay, so the rust, basically, the most of the interaction, it happens in the apoplast, so it's kind of a battleground. So, and these receptors, they are in the membrane. So these are the membrane localized. They have an extracellular domain that is open inside the extracellular spaces, that is the apoplastic spaces. And when this pathogen attack, it releases all kinds of PAMs, effectors, everything in that space. And that's why we actually utilize that space to take out the fluid so that there is a maximum chance to capture whatever the protein that interact or whatever the thing that interact with the LEC-RK, we can have it for our analysis. Yeah.
Awesome, thank you. And one more question from Lawrence Binchedler. Would LEC-RK also provide non-host resistance to cereal powdery mildews?
It has not been studied. We don't know, but yeah, it could be a good thing to test future.
Okay, we'll wait just a second and see if we get any other questions from the audience. And if not, oh, here we go. Zahur Mir asks that the study finds different recognition profiles among LEC-RK variants. This is scientifically interesting, but it complicates the claim that the mechanism is broadly conserved. I guess that's more of a statement, but can you address that statement?
The conserve is in a way like the GH5 and the LEC-RK perception. As we see, the GH5 were in the clade, which is highly conserved, and then we see that LEC-RKs, this interaction is still existing in the rice. We could see because we never know it was there if we would have not tested it. We tested other crops also. So when we took these crops, we could see that this is kind of broad, like even it can go to the other crops like rice and wheat. Yeah.
And then is the GH5 enzymatic activity needed for recognition by LEC-RKs?
No, it's not needed. We tested we actually created the catalytic mutants, and still they were able to bind, still they were able to trigger the HR, and yeah. So basically it acts as a BAMP without catalytic activity.
Okay. I think that is all of our questions. So thank you very much, Aruj. And now I would like to present our next speaker, Dr. Adrian Gonzalez, who currently works as a postdoctoral researcher at the University of Nottingham, but will be presenting a story that he published in 2024 during his PhD with Dr. Stephen Pullman at the Polytechnic University of Madrid. Today, Dr. Gonzalez will tell us about the molecular underpinnings of a beneficial plant-fungal interaction. So, Adrian, whenever you're ready, go for it.
Thank you so much, Abby, for the kind introduction. And thank you, everyone, for the invitation. It's a great pleasure to be here. And I want to talk about, as Abby has said, about a project that I finished a couple of years ago in Madrid. And to start with the story, I want to start talking about a global problem that we have. Nowadays, we are around 8 billion people, but it's predicted to reach almost 10 billion in 2050. Apart from this, the agricultural land use per person has been decreasing in the last decade. So to ensure food security to everyone, we have to produce more in less land. Apart from that, we also have the problem of climate change. As you can see in the graph, the global mean temperature has been rising up in the last years, which is increasing some abiotic stresses such as drought or salinity, which is decreasing crop yield. So the question here is, how are we going to produce more in less land, having this climate change problem in the future to ensure food security to everyone? As you may know, plants live in association with a wide variety of microorganisms, and it's been already tested. They have a huge impact on plant development. So for this reason, I think that to achieve the maximum improvements, the required innovations must take a holistic approach. In this story, I started studying the interaction between the model dicot plant Arabidopsis thaliana and this root colonizing endophytic fungus known as Serendipita indica, which was isolated in the Thar Desert in India. This fungus increases the plant yield and biomass production of its hosts, improves plant fitness and resistance, and which is very interesting, it has a broad host range. It is able to colonize, for instance, beetroot, tomato, rice, and even the model dicot plant, Arabidopsis thaliana. So as you can see in the screen, the Arabidopsis thaliana seedlings inoculated with the fungus has a huge impact on root architecture. And regarding this result, we started thinking that maybe oxygen homeostasis has a role on this phenotype. As you can see in the right part of the screen, this is the indole triacetic acid biosynthesis pathway. IAA is the main naturally synthesized auxin, and a previous result from JLab in Taiwan, they reported significant differences in Chinese cabbage inoculated with Serendipita indica of the final product, the two intermediates and the substrate of the IAA biosynthesis pathway. So concluding that the colonization of Serendipita indica has an impact on oxygen homeostasis on the host plant. As well, previous results from Alga Zuccaro's lab in Germany, they reported that the fungus is able to produce IAA from a free tryptophan pool, and they decided to develop fungal mutants without the ability to produce IAA, as you can see here, the TAM2-2, and they had an impact in the colonization at the early stages of the interaction. However, this ability to produce IAA doesn't interfere with the root growth promoting effect triggered by Serendipita indica, at least in barley plants. Finished with this introduction, in our lab, in a study led by Dr. Begoña Benito, they saw that the Arabidopsis thaliana seedlings inoculated with Serendipita indica had an increase in the rhizosphere pH at the early stages of the colonization. This result is important because IAA, when it's protonated, it goes into the plant cell through passive diffusion, whereas when it's deprotonated, it needs from active transport to enter into the plant cell. So regarding this introduction, the first question of my PhD was if the ability to produce IAA by the fungus is not the driver for plant growth promoting effect, what's the real driver of Serendipity indica mediated root growth promotion? To address this question, we decided to perform an RNA-seq analysis at 2 different time points. And our initial hypothesis was that maybe one of the genes related to IAA biosynthesis pathway in the host plant might be differentially expressed. However, we only see some of the tryptophan biosynthesis genes and some of the CYPs that are different related compounds biosynthesis genes were differentially expressed. The subsequent gene ontology analysis revealed that many co-terms were related to defense, which makes all the sense because at the end we are studying an interaction, although it's beneficial, between a fungus and a plant, and the plant tries to defend from the fungus. A previous result from our collaborators in Germany in Ralph O. Müller lab showed that this mutant superroot1-1 allele it has an auxin overproduction phenotype, a characteristic one, and when they inoculated with Serendipity indica, they were able to recover the wild-type phenotype. It was so interesting for us. However, this line has an impairment of the different response in the plant. However, I would like you to remind this result because it was important afterwards in the story. In the meantime, in Madrid, we were quantifying free IAA content and conjugated IAA contents. And as you can see, at 3 days after the inoculation, there is an increase in the free IAA that disappeared at 10 and 14 days after the infection. However, the IAA conjugated with aspartate has a complementary effect as free IAA. Our collaborators in Germany, they also tested the dual auxin reporter line to check auxin signaling in Serendipity indica inoculated seedlings, regarding that they have an increase in the auxin signaling in the first 24 hours in the inoculated seedlings. This result was also confirmed by us with the DR5-loop auxin signaling reporter line, but we also have seen an increase in the auxin signaling at 6 days after the inoculation. I'd like to say that IAA, when it's free, is biologically active, whereas when it's conjugated through the action of acylamido synthetases such as Gretchenhagen-3s, it's biologically inactive. As I mentioned before, the superroot1-1 mutant has an impairment in the different response, and for this reason, we decided to check one other auxin overproduction phenotype mutant, such as one of the overexpressor of the flavin monocygenases yucca. And as you can see here, this is a typically auxin overproduction phenotype. However, when we inoculated with Serendipity indica, we were able to recover the wild-type phenotype. With this result, we decided to run another RNA-seq analysis and contrast with the first RNA-seq analysis we have in Columbia plants. And the principal component analysis revealed 4 clearly differentiated groups. And after contrasting the data sets and comparing using Venn diagrams, long story short, we focused on this 402 genes group. Why this group? Because in this group, there contain the genes that respond to both high auxin levels and the co-cultivation with the fungus. The gene ontology analysis of this group revealed an overrepresentation of the response to auxin co-term, and inside this term, we found 33 genes. We could find a small auxin upregulated RNAs and several Gretchenhagen-3 genes. Among these GA3 genes, we decided to look for candidates. To select candidates, first, we were looking for those that were induced in the root, because it's the part of the plant which is colonized by the fungus. And the second condition was that this induction was during the earliest stage of the colonization. For this reason, we decided to check J34, J35, and J317 single knockout mutants. And what we found was that J35 and J317 single knockout mutant lost the ability, doesn't disappear, the beneficial effect of the fungus disappeared, suggesting that the loss of J35 and J317 interferes with the root growth promoting effect triggered by the fungus. To check that this phenotype is not due to the absence of the colonization, we also check through trypan blue staining both the roots of both single knockout mutants in chlamydospores and IFI associated to the mutant roots. To go further in this part, we also check the gene expression localization of both genes using YFP reporter lines in primary and lateral root tips, and what we found was a local induction of G35 and G317 in Serendipity indica inoculated seedlings. In the case of G35, the induction appears in the columella cells, whereas in G317 in the external layer of the lateral root cap. So apart from interfere with the lateral root growth promoting effect, it's also inducing locally the expression of G35 and G317. Thanks to the literature, we know that Serendipity indica is not able to colonize the meristematic and the elongation zone of Arabidopsis thaliana seedlings. However, we had an induction of G35 and G317 in a part of the plant which is not colonized by the fungus. So our next question was, what's the real signal which is inducing the expression of G35 and G317 upon Serendipity indica colonization? We already know that Gretchenhagen trees are induced by the accumulation of auxin, and to have an accumulation of auxin in the root tip, we started thinking about auxin polar transport. The pin protein. We were starting to think about the pin proteins. And we had here two different hypotheses. To create an auxin maximum in the root tip, we had two possibilities, or we had the increase of the basipetal transport mainly mediated by PIN1, or we had the reduction of the acropetal transport mediated by PIN2. Taking the first RNA-seq data, we saw a slight repression of PIN2, and for this reason, we decided to check PIN2 GFP reporter lines. And what we found was that the abundance of PIN2 was lower in Serendipity indica inoculated seedlings, suggesting that maybe this lower abundance might create an auxin maximum in the root tip that might be inducing G35 and G317. However, when we tried to publish this, one of the reviewers asked about the other pin proteins, the other long pins, those pins that are located in the plasma membrane, which are PIN1, PIN3, PIN4, and PIN7, because it might be a compensation between PIN2 reduction and PIN1. And we decided to check the other PIN GFP reporter line. And as you can see here, there is no significant differences between mock inoculated and Serendipity inoculated seedlings, suggesting that only there is a reduction in PIN2. To finish with this, we also found two different PIN2 mutants due to IER1 alleles that were isolated from a screening of root gravityprism. AR1-1 has a non-synonymous mutation in the 7th alpha helix, whereas AR1-2 has an early codon stop. And as you can see, the phenotype AR1-2 has a strong root gravityprism phenotype. But what was more interesting about this was that checking the gene expression of those lines without the fungus, we saw an induction of G35 and G317 in the weak allele of AR1-1, which basically is suggesting that maybe this small change in the protein structure might interfere with the auxin maximum creation in the root tip and might be inducing G35 and G317. We also checked the dry and fresh biomass of the seedlings, showing that we had a significant increase in root shoots and the whole seedlings, in particular in the weak allele of AR1-1. To finish with the story, I would like to summarize everything in two different phases. The first one is the establishment of the symbiosis, in which the fungus is able to provide IAA to start with the colonization, and a second phase, which is the root growth promoting mechanism phase, in which The fungus, we don't know how yet, is reducing the pin2 abundance in the root tips, which is creating an auxin maxima across the root tip and inducing the G35 and G317, which basically is triggering a fine-tuned auxin homeostasis control that promote the root growth promoting effect driven by Serendipita indica in Arabidopsis thaliana seedlings. I would like to say thank you to my lab mates. All the collaborators have been participating in this story, my two PhD supervisors, Stefan and Bego, and I want to give most of the acknowledgements of this story to Stefan, who has been supporting me since I started my PhD, and I'd like to say thank you and give him most of the recognition for this story. So thank you everyone for your attention, and I'd be happy to take any question. Thank you.
Great job, Adrian. Okay, so it looks like we do have one question for you so far. Zahur Mir asked, did you check the molecular mechanism involved in the interaction of GH3, GH3.5 with fungi, like in the PNAS paper, in which they found the trichoderma interacting with ABCB5 transporters and increased auxin efflux?
We didn't do that. We didn't check it because basically what we think is that the auxin conjugating protein G35 and G317 is a mechanism of control of the auxin maximum, and it's happening at root level, in the root tip level, because checking the bulk RNA-seq data, we have a slight reduction of, for instance, pin2 and a slight increase in the expression of G35 and G317, but we basically think that this is the point of control of the auxin maximum that Serendipita indica, we don't know how it's creating in the root tip.
Thank you. Okay, we'll give it a few more seconds just in case anyone has another question they want to put in the Q&A.
Or Abby, maybe I can ask a question.
Yeah, that'd be great.
Yeah. So yeah, great talk. Thanks, Adrian. I mean, there are many rhizosphere bacteria and also fungi produce IAA and auxin and other phytohormones. And how do you think you put it in the microbiome context of this auxin rewiring happening in natural system?
Yeah, I mean, this is even more complicated because if we have to put Serendipita indica into the microbiome context, I've read that, for instance, there is a bacteria of the taxa Barioborax that is able to try to control the auxin factor inside the root microbiome, which is, I mean, it's super complicated because, I mean, now we're studying just Serendipita indica with the Arabidopsis. But if you put even, for instance, bacteria that are consuming IAA or they're producing more IAA, and then you have another points of control during the interaction, which is even more complicated. So I don't think this is, I mean, I think this could be the starting point of the interaction, but we are not taking into account other factors that might be affecting, for instance, the contribution for the root microbiome. Because if you have a fungus that is able to produce IgA and then you have a disbalance of the root microbiome, probably you have even a detrimental effect instead of a beneficial effect. So I think it's more complicated than just what we have seen in the work today.
Excellent. And kind of in the same vein as Wenbo's question in terms of like interactions with other soil-borne microbes, Georgina asked if you have any idea whether Serendipita indica infected plants become more susceptible to soil-borne pathogens or root pathogens, etc.
I mean, I'm not so much into plant defense, but I think I've read that Serendipita indica colonization is also protecting for the plant of some pathogens. So might be competing with some of these pathogens in terms of root colonization and might be preventing for it.
Okay, excellent. And then I think we'll move on to our last speaker of the day. So thank you again, Adrian. Now I would like to introduce Federico Abajay, who is currently a fifth-year PhD student with Dr. Ezequiel Petrillo at the Institute of Physiology, Molecular Biology, and Neuroscience at the University of Buenos Aires. Federico completed his undergraduate degree at the University of San Luis before joining Dr. Petrillo's group, where he now works towards understanding changes in Arabidopsis gene expression and how these changes influence plant immune outcomes. So, Federico, whenever you are ready, feel free to take it away.
Thanks for the introduction. Can you see my screen?
Yes.
Okay, cool. Thanks for the introduction. I am Federico, and today I will present to you my work titled Alarmon Signaling Tunes the Nuclear Transcriptome to Control Arabidopsis Defense Responses. So, gene expression in eukaryotes is a complex process that involves many regulatory layers. Among these layers, we can find the communication between the nucleus, between the nuclei and the plastids. One of the signals that mediate these communications are the signals, that mediate this communication is the anterograde signals. And an easy example of these signals can be the genes that are encoded in the nucleus that are transcribed and then get translated, and the protein product goes into the chloroplast. And for example, we can find into the photosystem 2, photosystem 1, and the ATP synthase, different nuclear encoded proteins and chloroplast encoded subunits. So in the other way around, we can find the retrograde signals. These signals can be the tetrapyrols, the reactive oxygen species, among others. So among these signals, we can also find, or in the world of these signals, we can find the guanosine pentaphosphate or guanosine tetraphosphate, also known as alarmon. The alarmon is a hyperphosphorylated nucleotide that has 4 or 5 phosphate groups, and its metabolism is controlled by the RSH enzymes. RSH1 is a hydrolase, RSH2 and 3 are both hydrolase and synthase, and the CRSH is only a synthase. And these RSH enzymes are very special because they are like dephosphokinases enzymes, which is a very specialized types of kinase. So, among the alarmon known functions, we can find the regulation of the chloroplast function and size, the regulation of the photosynthetic efficiency in a torr-dependent manner. This molecule can also relieve the plant stress to nitrogen deficiency and highlight, and it can also regulate the leaf senescence. As an example of this, we can find that plants with high levels of alarmon exhibit smaller chloroplasts and a higher number of them. So the main objective of my PhD is to find out whether the alarmon is a novel plastid-based signal able to regulate the nuclear transcriptome. To this end, we performed an RNA sequencing of Arabidopsis seedlings of the wild type, the RSH3 overexpressor line that has high levels of alarmon, and the RSHQ line that has no levels of alarmon. To mention, the RSHQ line is a mutant that has all the RSH enzymes mutated. So, by doing a geo-analysis, we found many immunity-related terms in the RSH3 line. And for the case of RSHQ line, we also found other immunity-related terms, and also we found terms that are related to the secondary metabolism that is linked to immunity. So, in the case of the plants with high levels of alarmon, if we take a further look at some of these genes, we can find BSMT1, which is responsible for the methylation of salicylic acid, JOX3 and 4 that are responsible for the hydroxylation of jasmonic acid, and HCA1, which is a jasmonic acid or a susceptibility marker. And for the case of the RSHQ line, or when you have low levels of alarmon, we can find a mild expression of PR1 and PR2. and PDF 1.2a and PDF 1.3. And just to remember, PR1 are salicylic acid markers and PDF are jasmonic acid markers. So the alarmon is somehow regulating the expression of these genes that are nuclear encoded and that are linked to the immunity. So we know that the alarmon is regulating the expression of immunity genes, but is this molecule really produced under infection conditions? So to this end, we infected Arabidopsis seedlings with Pseudomonas syringae DC3000 and measured the expression of the different genes at different time points. For the case of RSH1 and CRSH, we didn't find any change, but for the case of RSH2 and 3, we found that the expression of these two genes is increasing along throughout the different time points. So since these two genes are both synthase and hydrolase, we've got evidence that can tell us that these genes in this moment are acting as synthase enzymes. So to further understand this phenomenon, we infected the plants with the Delta HRC strain of Pseudomonas, which is a non-pathogenic strain that cannot inject its T3SS effectors due to a mutation in one of the genes that encode the proteins for this complex. And we found that none of these genes are being affected, especially RSH2 and 3, where we previously saw the change. So the T3SS activity is necessary to induce the expression of these genes. So what happens when the plants have different levels of alarmon and infection? So we first infected the plants and measured the bacterial growth at different time points. And when we checked the bacterial growth of the plants with high levels of alarmon, we could already see the hypersusceptibility phenotype at 24 hours post-infection. And this phenotype gets exacerbated throughout the experiment. And for the case of the null levels of alarmon, the plants have a little bit lower levels of alarmon, and we can find the resistant phenotype at 48 hours post-infection. When we see the disease symptoms, we can find some symptoms in the cold zero plants, of course, and for the case of the high levels of alarmon, we can see that these symptoms are stronger. But for the case of the RSHQ line that doesn't have any alarmon, we don't see any symptoms, or we don't see any apparent symptoms. So we can say that the plants with high levels of alarmon exhibit hypersusceptibility, and the lower levels of alarmon promote their resistance to Pseudomonas. So to understand the molecular basis of this phenomenon, we first checked the expression of PR1, which is normally activated during pathogen infection. However, for the case of the RSH3 plants, we didn't observe an increase in the expression of this gene, meaning that probably the hypersusceptibility phenotype might be explained to an abolished salicylic acid signaling in these plants. And for the case of PDF 1.2, we also measured the expression of PDF 1.2, which is a jasmonic acid signaling marker, and observed that this gene is, the expression of this gene is actually exacerbated in the plants with null levels of alarmon. So maybe the resistance against Pseudomonas in these plants might be due to an exacerbated jasmonic acid signaling. Moreover, we checked the expression of a PTI marker, which is FRQ1. FRQ1 is activated due to the pathogen sensing, and the expression normally goes up in the wild-type plants, and we can see the same effect in the RSHQ plants. But for the case of RSH3 plants, we didn't observe an increase in the expression. So if we take a look at one of the PTI responses, which can be the stomatal immunity, we observe first that the stomata are being closed during pathogen exposure for the case of the cold zero plants. And we observe the same phenotype for the case of the RSHQ plants. So we don't observe any difference in the case of null levels of alarmon. However, when we checked the plants with high levels of alarmon, we can see that first thing first, they got a wider stomatal aperture, even in normal conditions. However, when these stomata are exposed to Pseudomonas, these stomata are not being properly closed. So the high levels of alarmon are affecting the stomatal immunity of the plants. So I previously mentioned that alarmon regulates the chloroplast function and the photosynthetic efficiency. So the photosynthetic efficiency, just to give you a short introduction, I want to say that the light energy absorbed by the chlorophyll in plants can be can excitate the chlorophyll, and this energy can be used as heat, can be dissipated as heat, can be re-emitted as fluorescence, and can be used to drive the photosynthesis. And we can measure actually the fluorescence that these plants emit. And the measurement of the chlorophyll fluorescence is a measurement of the photosystem 2 activity in response to a certain stimulus. And this is what we are going to call photosynthetic efficiency. So the plants with high levels of alarmon normally exhibit a lower photosynthetic efficiency in normal conditions compared to the wild type. So if we measure the photosynthetic efficiency upon pathogen infection, I want to first mention that this is normalized to the time zero, and the mock treatment does not affect the photosynthetic efficiency. When we infect the plants with Pseudomonas, wild type, we observe a downregulation in this parameter, and this effect is stronger in the plants with high levels of alarmon. However, for the case of the RSHQ line, we do not observe any change in the photosynthetic efficiency. And we can see it here that the photosynthetic efficiency is going down in the infected leaves, and we don't see any change in the RSHQ. However, in addition, we also infected the plants with the Delta HRC strain. And we didn't observe any change in the photosynthetic efficiency, even in the plants with high levels of alarmon. So we can say that first, the alarmon is regulating the photosynthetic efficiency during pathogen infection, and this phenomenon is induced, let's say, by the T3SS system. So we have now checked, I have talked to you about the hemibiotrophic pathogen Pseudomonas, but what happens with the necrotrophic pathogen botrytis? So just to mention that in the RNA sequencing data, we found some genes that are involved in the resistance against botrytis. So we decided to infect the plants and we decided to inoculate the plants with mycelium of botrytis and measure the the mycelium invasion size at different time points. And at 72 hours post-infection, we can find that for the case of the RSHQ line, they exhibit a so much higher lesion size, mycelium invasion size, compared to the wild type. And we can see a mild effect on the case of the RSH3 line. So if we measure the expression of PDF 1.2, which is a very well-known botrytis resistance marker, we can see that the expression of this gene is being upregulated in the wild type as normally. We don't see any change for the case of the RSH3, We also saw this effect on the case of Pseudomonas. However, for the case of the RSHQ line, we don't see any change in the expression of PDF 1.2. And this might be explaining first this phenotype of the hypersusceptibility. But this is still striking to us because this gene was being strongly expressed during Pseudomonas infection. So there might be a different jasmonic acid pathway signaling activated during botrytis infection in these plants. We also checked the photosynthetic efficiency in these plants, and we first observed that the plants inoculated with botrytis are suffering, let's say, in the photosynthetic efficiency because this parameter is going down around the infection site of botrytis, and this effect is stronger in the RSH3 plants. However, for the case of the RSHQ plants, we don't observe any change in the photosynthetic efficiency around the infection site, and this is, and we are still seeing the dead tissue because we see the white pixels here. So this is giving us the idea that the alarmon is strictly regulating the photosynthetic efficiency during infection. So to conclude with these results and a couple of them that we have in the paper, we can say that the alarmon levels can modulate the jasmonic acid or salicylic acid balance. The high alarmon levels promote susceptibility to Pseudomonas, while low levels promote resistance. The salicylic acid pathway is required for the expression of RSH2 and 3. Alarmon is required for the resistance against botrytis, and this molecule strictly regulates the photosynthetic efficiency, and in the case of Pseudomonas, in a T3SS activity-dependent manner. So I want to finish, I've got some unanswered questions that we still have. How is botrytis killing plant cells without affecting chlorophyll fluorescence? What is the basis of RSHQ plants' resistance to Pseudomonas? And what are the specific pathogen effectors that are affecting alarmon levels? So I do want to finish now this arrow should not be here. I don't know where it's here. I want to thank my team, Rocio, Flor, and my wonderful supervisor that really supports me. And he's the creator idea of this project. So thank you for this opportunity.
All right, great job, Federico. We do have a couple questions, but before I get to those, I do want to mention, as we come up to the end of this webinar, as it ends, all of the audience members should be prompted to fill out a survey. So if you could please fill that out, that would be very helpful for us. We do have a question here from Zahor Mir, who asked, how does the alarmone regulate PR1 and PDF gene expression? And then did you check the expressions of MYC2, 3, and 4, as they are the main signaling genes involved in the JA pathway during biotic stress?
Okay, thanks for this question. How alarmon is regulating the expression of PR1? Sorry, give me just a second because I'm having an issue with the battery of my computer. I don't know why. Okay, so we don't know how alarmon is regulating the PR1 expression because we don't know how this molecule from the chloroplasts regulate the expression of nuclear genes. We don't have that information yet. And nope, I didn't check the expression of MYC2 and the others, but it will be interesting to do it.
Okay. Excellent. And then we have one here from Abin Panakal George, who this is quite long, so I'll read it slowly. And if you maybe want to jump in to answer as I'm going or address any of the statements made, that might be helpful just to kind of answer as we go along. So they've said, we observed an induction of JA and oxylipins in the RSH overexpression lines. However, the transcription of JA responsive genes was increased in the knockout lines. Interestingly, despite the elevated levels of JA and oxylipins, which are generally associated with defense responses, the RSH overexpression lines were more susceptible. This raises the question of why increased accumulation of JA and oxylipins in the overexpression lines does not result in enhanced defense, but instead is associated with increased susceptibility. Furthermore, many JA responsive genes, including PDF 1.2, or maybe perhaps that's 1 and 2, are well known to contribute to defense against necrotrophic pathogens. However, previous studies have shown that PDF 1.2 is strongly induced during Pseudomonas infection, but not during infection by botrytis. This suggests that JA signaling and its downstream transcriptional responses are highly context dependent. It would be interesting to understand how JA responsive genes are differentially regulated. So that was the entirety of the sort of thought there. If you need me to repeat any portion, please let me know. I guess, how would you think to address this?
So can you still hear me?
Yes.
Yes.
Okay. So yes, I have thought about that, and this has given me a lot of headache. Yes, we observed like a higher expression of JA-related genes in the RSHQ line, but we think that under infection there is something else going on in these plants. Just to give you an idea of what might be happening, we think that these plants have how do I say it in English? A higher susceptibility to death, earlier than other plants. I mean, they probably have an I forgot the word, hypersensitive response, and that might be let's say, the reason why they are being more resistant to Pseudomonas. Because the Pseudomonas is actually a hemibiotrophic pathogen, so in the first phase, it needs live cells. And that's it. That's the... So as the lack of alarmone are inducing the early cell death response, probably botrytis is not contributing, but it's like the plant is giving a favor to botrytis to have I don't know how to say it. It's like the plants are ready to botrytis to proliferate more, since the plants can die easier, let's say. I don't know how to say it in English, sorry, but I hope the question has been answered. You can always reach out to me.
Awesome, thank you. And we do have one other question from Ritzko Hadi. Does alamone, is it also involved in responses to other pathogens like viruses?
Yes, there is a paper from 2018 where one of the collaborators from us reported the role of alamone in response to turnip mosaic virus, and they observed the same effect that we observed in the case of Pseudomonas. Null levels of alamone promote resistance to DMV, and high levels of alamone promote hypersusceptibility to the virus.
Excellent. Thank you very much, Federico. And I think that is all the questions we have for you. So I guess Jean or Mauricio, you want to close out the seminar?
Thanks everybody for participating, and we look forward to seeing you at the other Road to Jeju events. Please answer the survey. And with that, I'll say thank you for participating. Thanks to all the speakers. Yay.
Thank you. Thank you all.
Yeah, so, okay, gonna sign off now. Bye, everyone.